Good morning or good afternoon, everyone. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is A Multiomics Approach to Defining the Nature of Early Malignant Transformations of the Immune System. The sponsor of our webinar is BD. Our speaker today is Wendy Béguelin, Assistant Professor of Pharmacology and Medicine at Weill Cornell Medical College. You may type in a question at any time during the webinar, and you can do this through the control panel, which normally appears on the right side of your screen. Just click on the Q&A box in the control panel, and please make sure the drop-down menu says All Panelists. In case you don't see the Q&A box in the control panel, just move your cursor a little over to the left over the main panel until you see the control icons on the bottom, and then go to the Options menu and select Q&A. We will ask our speaker your questions after her presentation has concluded. So let me turn it over to Wendy. Please go ahead with your presentation.
Hello, everyone, and thank you for attending this seminar. This is the outline of the seminar. I'll give you a brief introduction to the germinal center reaction and the role of the enzyme EZH2. I'll show you what we knew about mutant EZH2 before performing single-cell RNA-seq, our experimental approach using BD Rhapsody, how we analyzed our targeted single-cell RNA-seq data, our findings in mutant EZH2 germinal center B cells, and finally, some other experiments that were triggered by the results obtained from targeted single-cell RNA-seq. So first, an introduction to the germinal center reaction. During the humoral immune response to infection, subsets of mature resting B cells are stimulated to migrate to the lymphoid follicles located in lymph nodes and spleens. Within lymphoid follicles, activated B cells form germinal centers, where they proliferate, mutate their antibodies, and differentiate into memory B cells or antibody-secreting plasma cells. The function of these differentiated B cells is to generate high-affinity antibodies. Germinal center B cells are a very complex mixture of cells undergoing several different transitional states. In the germinal center dark zone, B cells, called here centroblasts, undergo massive proliferation and clonal expansion, along with simultaneous somatic hypermutation of their immunoglobulin genes. Once these centroblasts have stopped proliferating, they are known as centrocytes, which are located in the light zone of the germinal center, and they are subjected to selection by follicular helper T cells in the presence of follicular dendritic cells. Negatively selected centrocytes undergo apoptosis. Positively selected centrocytes might recycle to centroblast and re-enter the dark zone for additional cycles of somatic hypermutation and division. And only the few B cells that develop mutated immunoglobulin genes with high affinity for antigens are allowed to survive and differentiate to plasma cells or durable memory B cells. Principal component analysis of the transcriptome of mature B cells at different stages of differentiation shows a clear segregation of B cell subsets. The first principal component here distinguished naive B cells, centroblasts, centrocytes, and memory B cells from plasma cells, whereas the second principal component distinguished naive and memory B cells from centroblasts and centrocytes. A phylogenetic analysis of the transcripts shows that the phylogenetic distance between centrocytes and memory B cells is similar to the distance between centrocytes and centroblasts, and it's shorter than the distance between centrocytes and plasma cells. It also suggests that the germinal center B cell transcriptome represents a departure from the B cell differentiation program, which is restored after exit from the germinal center reaction and before terminal differentiation. Understanding the biology of the germinal center reaction is very important because germinal center B cells give rise to the great majority of B-cell lymphomas, including follicular lymphomas and diffuse large B-cell lymphomas. Germinal center B cells upregulate EZH2. Although it's silenced in resting naive B cells, it's highly induced in germinal center B cells. EZH2 is a core component of polycomb repressive complex 2 that methylates lysine 27 hosting 3 to generate H3K27 trimethylation, a histone mark associated with gene repression. EZH2 is absolutely required to form germinal centers. Conditional deletion of EZH2 results in failure to form germinal centers. And its knockdown or specific EZH2 inhibitors suppress B-cell lymphoma growth. Approximately 30% of patients with the GCB subtype of diffuse large B-cell lymphoma and follicular lymphoma have gain-of-function mutations of EZH2. Mutant EZH2 drives aberrantly efficient trimethylation of lysine 27 histone 3, while wild type is more efficient to catalyze the monomethylation of H3K27. So to study the impact of the lymphoma-associated gain-of-function mutation of EZH2 in vivo, we generated a mutant EZH2 conditional knock-in mouse model where the point mutation tyrosine 641F was introduced in the endogenous EZH2 locus. In absence of CRE-recombinase, wild-type EZH2 is expressed from the mini gene. When CRE-recombinase is present, the mini gene is deleted and mutant EZH2 is expressed. We crossed this model to C-gamma1 CRE mice to induce expression of mutant EZH2 specifically in germinal center B cells. And we further crossed these mice with YFP reporter mice, where a stop flux cassette allows the expression of YFP in CRE-positive cells. So YFP is expressed in germinal center B cells and post-germinal center B cells. Mutant EZH2 mice develop hyperplasia of germinal centers, which eventually results in lymphomagenesis. So with these tools, we decided to explore the mechanism through which EZH2 mutations disrupt the immune system to initiate malignant transformation. We first did the histone mass spec in purified germinal center B cells, and we found a massive increase in H3K27 trimethylation levels in mutant EZH2 cells at the expense of reduction in dye and monomethylation. ChIP-seq experiments in purified centroblasts and centrocytes consistently show the huge gain and spreading of H3K27 trimethylation in mutant EZH2. And as expected, most differentially expressed genes were downregulated in mutant EZH2 centroblasts and centrocytes. Among them, we found genes encoding for proteins involved in interaction with T cells like MHC class II, IL-21 receptor, ICAM1, and the pathways that mediate recycling of centrocytes back to the dark zone, including some MYC target genes. So we decided to study the transcriptome of the normal germinal center reaction and alteration caused by the lymphoma-associated mutant EZH2 at a single cell level. And why BD rhapsody? So germinal center B cells are a very complex mixture of cells undergoing several different transitional states, as I explained in the introduction. Single-cell transcription analysis is the only way to precisely tackle how mutations occurring in germinal center-derived B-cell lymphomas affect these states and eventually cause cell transformation. Global single-cell RNA-seq cannot mechanistically resolve these cell transitional states, since the most represented genes are not necessarily the most relevant genes. So we reasoned that BD rhapsody would enable such fine-tuning of transitional state characterization within the germinal center reaction. Okay, so our experimental plan. We immunized 3 wild-type and 3 mutant TCH2 mice to allow germinal center formation. We then took spleens and lymph nodes. And we sorted YFP positive cells, which includes germinal center B cells, memory B cells, and plasma cells. We decided to use the YFP Cree reporter system to avoid the sorting based on specific surface markers of predefined populations. I want to clarify that we didn't do AppSeq for this project. We also sorted few T follicular helper YFP negative cells. And post-sorting, we mixed 95% YFP positive cells with 5% TFH. And we tagged the 12 samples with the BD single-cell multiplexing kit. Then we pulled the tagged samples and split them into 4 cartridges. We loaded 20,000 cells per cartridge. And then we followed the Rhapsody protocol. We loaded the cell capture beads, lysed and retrieved beads from cartridges, then reverse transcription. And for PCR, we used the immune response panel, which targets 400 genes approximately. Later on, but not for this particular experiment, we had our own custom panel design, where we added 100 genes relevant for germinal center biology. So here you have the flexibility of adding your custom panel design or even go full custom, up to 500 genes. Then library prep, and for sequencing, we used 1 lane of NextSeq 500, and we did pair-end 75 base pairs. We analyzed the data using BD data view software. Okay, out of 20,000 loaded cells, we captured an average of 4,000 per cartridge. We could detect a very good number of genes, 388 out of 397 in file. We detected a median of 552 RNAs per cell with a minimum of 26 and maximum of 22,000. And the median gene per cell was 84 with a minimum of 7 and maximum of 201, which of course is expected because of the heterogeneity of the cell populations. 1 lane of NextSeq 500 generates 500 million reads, which in this case was enough because 94% were passing genes. This is successfully detected. But in this example, in a different experiment, 1 lane of NextSeq 500 was not enough. The proportion of reads from low-def genes was 46%. After a second round of sequencing, the panorama improved significantly. Now, 94% are passing genes. Looking at the distribution of the TSNP maps per cartridge, we didn't observe any batch effect caused by different cartridges. So this is how specific gene expression looks in the TSNP plots that we made with the BD data view. I'm showing here C19 to highlight the B cell clusters. And as a comparison, we can also distinguish this population by flow cytometry based on surface protein expression. And then a different population, CD4 positive cells, highlights the T helper, the T helper cluster on the top. By traditional flow cytometry gating, we can also distinguish these populations based on surface markers. I'm showing here some examples of gene expression at the top and protein expression at the bottom of markers that distinguish subset of B cells. CD38 shows the non-germinal center B cells. CD86 shows the light zone within germinal center B cells. And CXCR4 on the right side is a typical marker for dark zone, which in this case, either the gene was not differentially expressed or the probe didn't work. This is a case where 2 different probes were designed to target the same transcript, SNC3, and one of them didn't work. So the correlation coefficient is, of course, very, very low. In this case, the correlation between two different probes for S1PR1 was very good. So a great advantage of targeted single-cell RNA-seq is that we can target different isoforms of the gene of interest. We then tried to compare wild-type splenocytes from two independent experiments, and we found a massive batch effect. So do not compare samples from different experiments. If you have to run the experiment separately, you will find a safe stopping point in the Rhapsody workflow. This is after the PCRs and before amplifying the libraries. Okay, how to define the clusters? And the million-dollar question, what is a good number of clusters? The clustering algorithm is based on hierarchical clustering and identifies statistically significant clusters. DataView software has two clustering methods available, both based on the dendrogram. Maximum cluster number on the left side, where you can define the number of clusters based on what you know about the biology of the populations, and recursive dendrogram split on the right side will give you different optimal number of clusters. So the criteria for choosing the number of clusters can be both mathematical and biology based. I just wanted to clarify that our Disney plots here look different because we decided to perform the analysis with splenocytes only, excluding lymph node cells. The reason of this is that the immunization time point we used was ideal for splenic germinal centers, but already too late for lymph node cells. To identify the clusters, we can look at the expression of the gene sets. BD DataView have some predefined population signatures, shown here on the left side as an example, pan T cells. You can clearly see the T cell cluster here. And you can also project your signatures, as shown on the right side for germinal center lysosomal genes. So it's a good tool to incorporate bulk RNA-seq data. Now, you can use the sum of multi-gene expression, as shown in this slide, which displays the sum of the number of molecules from all genes in highlighted gene set detected per cell, or the number, which displays the number of genes in the highlighted gene set detected per cell, or the proportion, which is the sum of the number of molecules from all genes in the highlighted gene set divided by the sum of the total number of molecules per cell. In the case of our germinal center lysosome gene set, shown on the right side here, the proportion of multi-gene expression highlighted the lysosome cells better than the sum or the number of multi-gene expression. Another way to identify the clusters is by differential expression analysis. You can compare differentially expressed genes in one cluster versus the rest, as shown on the left side, or compare two individual clusters, as shown on the right side of the slide. This tool is especially useful for close clusters that don't separate in the Disney map. And the third and actually very easy way to identify clusters is by displaying individual gene expression. We can look at the expression of known hallmark genes, as I showed before for CD19 and CD4, or in this example for AACDA, a germinal center hallmark gene. In all this expression analysis that I showed, we can look at the gene expression by genotype to compare wild type versus mutant TCH2. Okay, so using a combination of tools to identify clusters, we could identify T helper cells, B cells, plasma cells, germinal center B cells, and memory B cells. Within germinal center B cells, we could easily distinguish centroblasts in the dark zone from centrocytes in the light zone. But not only that, because the centroblasts are highly proliferative cells, we noted that they were also clustered by cell phase stage. In summary, we identified 11 different clusters in our YFP-positive splenocytes: T helper cells, plasma cells, plasmablasts, which are proliferative, less differentiated plasma cells, memory B cells, committed centrocytes, centrocytes, recycling centrocytes back to dark zone, centroblasts transitioning to light zone, centroblasts in S phase, and centroblasts in M phase. And consistent with the phylogenetic analysis of the transcripts that I show in the introduction, memory B cells are found in the same big cluster with centrocytes, and the plasma cells form a totally separate cluster. Next, we wanted to focus on germinal center B cells. So we removed T cells and plasma cells, and then reclustered the germinal center and memory B cells. Because we observed a clear reduction of mutant TCH2 recycling cells, we decided to investigate further these cells. The proto-oncogene MYC is shut down in the dark zone, but it's briefly and strongly induced in centrocytes selected for dark zone re-entry. So to identify and track these recycling B cells, we used a MYC-reported mouse model where the endogenous MYC encodes the GFP-MYC fusion protein. We crossed them with wild-type and mutant TCH2 mice carrying C-gamma1 Cre. We immunized them to allow germinal center formation, and 8 days later, we harvested the splenocytes for flow cytometry. Consistent with targeted single-cell RNA-seq data, we found a severe reduction of GFP-positive germinal center B cells in mutant TCH2 mice, indicating a failure of mutant TCH2 centrocytes to re-enter the dark zone. Also consistent with the Rhapsody data, GFP-MYC positive cells are exclusively centrocytes. This is CD86 high, CXCR4 low. Another population that had clear proportional changes when we compared wild type to mutant TCH2 in the germinal center Disney plot was the centrocytes, the light zone. Mutant TCH2 was increased in clusters 6 and 7 taken together. So we evaluated the proportions of centroblasts and centrocytes in wild-type and mutant germinal centers by flow cytometry using the surface markers CXCR4, CD83, and CD86. And we confirmed the expansion of centrocytes in mutant TCH2 mice. While the ratio in normal mice for centroblast centrocytes is usually 2:1, mutant TCH2 germinal center B cells have a ratio closer to 1:1. We also confirmed the expansion of the light zone in mutant TCH2 mice by immunofluorescence confocal microscopy of spleen sections. This is how germinal centers of YFP reporter mice look under the microscope. The pictures on the top are wild-type EZH2 and on the bottom mutant EZH2. We stained the follicular dendritic cells with anti-CD35 antibody in blue to highlight the light zone. And you can clearly see the expansion of the follicular dendritic cell network in mutant EZH2 germinal center. Our next question was if there are differences in the proliferation of mutant EZH2 germinal center B cells that would also explain the light zone expansion. The analysis of Rhapsody didn't show significant differences in wild-type versus mutant proliferating cells. So we decided to address this question with a different mouse model. We used the FUXI2A strain that encodes probes that fluoresce red during G1 because it's cherry, and green during S, G2, M phases because it's Venus. This allows the study of the cell cycle dynamics. We determined the gating strategy of the different phases of cell cycle in Venus versus cherry flow plots based on the germinal center B cells and naive non-germinal center B cells. As expected, most germinal center B cells are found in S, G2M phases. This is cherry negative, while naive B cells are in G1 and G0. This is cherry positive, Venus negative. On the right side of the slide, you can clearly see in the microscopy picture germinal center cells in green or colorless, which are late M, early G1, and the naive B cells surrounding the germinal centers are red. We validated our gating by evaluating DNA content using ZAPI in fixed Fuchs splenocytes, where ZAPI shifts to the right in cells in earliest, and the shift is more pronounced in cells in SG2M. When we gated in centroblasts and centrocytes, we found most centrocytes in G1 and centroblasts in SG2M phases as expected. When we compared wild type to mutant TCH2 Fuchs splenocytes, we found a significant increase of mutant TCH2 centrocytes in SG2M phase and decrease in G1 phase. These differences were not striking in centroblasts, which are the most proliferative cells in the germinal centers. And this is why we think we couldn't find differences when we analyzed the Rhapsody data. Confocal microscopy images of the mutant TCH2 Fuchs spleen show green proliferating cells in the light zone, the picture on the right. And the light zone here was identified by CD35 staining in blue, while the wild-type TCH2 germinal centers, the pictures on the left, these green cells are mostly restricted to the dark zone. Okay, next we focused on plasma cells. From the original T-SNP plot, we observed a clear increase of mutant TCH2 plasma cells. After removing T cells, germinal center B cells, and memory B cells, we identified 8 clusters, which main differences were the immunoglobulin isotypes. Among them, we found significant differences between wild-type and mutant TCH2 proportional changes, being IgA more enriched in wild-type TCH2 and IgG1 more enriched in mutant TCH2. This is how the T-SNP plot looks after reclustering, and the distribution of wild-type and mutant TCH2, where we can see more clearly the differences in immunoglobulin isotype. It is also evident that mutant TCH2 produces more plasma cells. If we didn't have the information of immunoglobulin genes, everything would cluster together. And this is actually what we observed when we did a global single-cell RNA-seq, where we didn't have Ig gene expression. We confirmed the expansion of plasma cells in mutant TCH2 mice by monofluorescence confocal microscopy of spleen sections. The bright YFP cells outside the follicles are plasma cells that we stained with CD138 in red. To evaluate Ig production in wild-type versus mutant TCH2 mice, we did ELISpots with splenocytes and bone marrow cells, and ELISA with serum from immunized mice. ELISpot assays confirmed that mutant TCH2 plasma cells produce higher amount of IgG1. And the ELISA confirmed that wild-type TCH2 produce higher amounts of IgA at short but not long times of immunization. We are now repeating this experiment to confirm the results. Based on B cell population shift that we observed, and also based on gene expression patterns, we hypothesized that EZH2 mutation makes B cells less dependent on T cells. So we first checked the surface molecules involved in interaction with T follicular helper cells. Although we've seen increase of H3-ketogen trimethylation in genes encoding some of these receptors in the mutant TCH2 centrocytes, So we were expecting these genes to be more repressed. We didn't find a significant decrease in protein expression by flow cytometry. So we decided to block the interaction between germinal center B cells and the T follicular helper cells in vivo using an anti-CD40 ligand antibody. Treatment of control mice with anti-CD40 ligand antibody inhibited the germinal center reaction, but it didn't significantly affect mutant EZH2 germinal centers. To validate these results in a context where wild type competes with mutant EZH2 in the same microenvironment, we did a mixed chimera bone marrow transplantation experiment. As wild type bone marrow donor, we used a mouse carrying the differential panleukocytic marker CD45.1, and mutant EZH2 expresses the CD45.2 allele. This is how we can distinguish them by flow cytometry. We mixed 50% wild type and 50% mutant bone marrow and injected into irradiated RAG1 knockout host mice that produced no mature T or B cells. 1 month after transplant to ensure engraftment, mice were immunized and treated with anti-CD40 ligand antibody. Mutant EZH2 germinal center B cells had a competitive advantage over wild type in expanding the germinal center. And this advantage was even greater when we blocked B cell T cell interactions, suggesting that EZH2 mutation makes germinal center B cells less dependent on T cell selection. This evidence of a differential selection pressure in the light zone in presence of EZH2 mutation prompted us to evaluate the clonality of mutant EZH2 germinal center B cells. For this, we crossed the mutant EZH2 mice with the stochastic multicolor Cre recombinase reporter Confetti or rainbow mice. This model allows a way to label and distinguish individual cells with 4 possible outcomes after Cre recombination: nuclear localized GFP, membrane target CFP, cytoplasmic RFP, or cytoplasmic YFP. And in homozygosis, we have 10 possible combinations. So when crossed to sigma-1 Cre, we can label individual germinal center B cell clones. We found that mutant EZH2 germinal center B cells, pictures on the bottom, established rapid clonal dominance patterns in contrast to the wild type, which are the pictures on the top, which only develop clonal dominance late in the germinal center reaction and only in a subset of germinal centers. The quantification of Confetti confocal images on the right side of the slide shows a reduced Shannon entropy in mutant EZH2 germinal center B cells. This means that there were less combination of colors. In other words, less clonal diversity in mutant EZH2. Finally, we evaluated the clonality at a genomic and single cell level. For this, we vibraton cut 3 wild type and 3 mutant EZH2 YFP fresh spleens. We manually cut off individual germinal centers that were identified by fluorescent microscopy. And each germinal center was stained with dark zone and light zone germinal center B cell markers, and single cell indexed sorted into 96 well plates. Then we isolated RNA, reverse transcription, and we PCR the IgG and IgM loci. Next, barcoding, indexing, library prep, pooling, and finally sequenced using my seq pair end 300 base pairs. Exactly the same process was done with the Confetti spleens. And this is my last slide of results. we found a reduced number of mutations in mutant EZH2 IgG1 and similar mutation burden in IgM in both YFP and Confetti mice. And the reduced diversity of clones in mutant EZH2 mice with a clonal dominance in the light zone, as shown in the phylogeny trees of the clones on the lower right side of the slide. This light zone clonal dominance in mutant EZH2 mice further support the results that these centrocytes are not re-entering the dark zone. So, what we learned from targeted single-cell RNA-seq. If we go back to the phylogenetic analysis of the transcripts obtained by bulk RNA-seq of sorted B-cell population that I showed in the introduction, and we add what we learned from targeted single-cell RNA-seq analysis using BD Rhapsody, we can insert now new transitional subpopulations with different biological roles within the germinal center. This analysis also determined the proportional changes of these populations in mutant EZH2 cells. Triggered by these targeted single-cell RNA-seq results, we also learned a lot of things about the molecular mechanism through which EZH2 mutations disrupt the immune system to initiate malignant transformation. Low-grade follicular lymphomas are tumors consistent of slowly proliferating light zone germinal center B cells. Given that centrocytes normally recycle to the proliferative dark zone, it is hard to imagine how such tumors could ever form, since centrocytes should be quickly overwhelmed. Our study provides the first evidence for how this might happen, whereby mutant EZH2 causes centrocyte hyperplasia, accompanied by expansion of follicular dendritic cells, shutdown of the germinal center recycling from light zone to dark zone, aberrant proliferation of centrocytes in the light zone, and the expansion of the IgG1 plasma cell production with clonal dominance and independence of T cell selection. So our dissection of the mechanism of action of mutant EZH2 provides the first model that reflects the biology of low-grade lymphoma. With this, I want to thank all the people involved in the project, especially Leandro Venturuti, who helped design and perform the Rhapsody experiment, and Christopher Chin, who analyzed Rhapsody data with the DataView. I also want to thank our collaborators, the people from BD Biosciences that helped us with all the Rhapsody procedure, and the funding agencies. And thank you for attending this webinar. I'll take any questions. Julia?
Thank you, Wendy, for your presentation. As a quick reminder, if you have a question, please type it into the Q&A box. And once again, if you do not see the Q&A box in the control panel, just move your cursor to the left over the main panel until you see the control icons on the bottom, and then go to the options menu and select Q&A. Also, just before we start with the Q&A, I'd like to ask our attendees to take a very brief moment after the webinar has ended to take our exit survey and provide us with your feedback. So with that, let's move into the questions. So for the Q&A, we have two people joining us, Leandro Venturuti, a postdoctoral associate in medicine at Weill Cornell Medicine, and Christopher Chin, a computational biology and medicine graduate student also at Weill Cornell, and they were both acknowledged before by Wendy. So let me see what we have in terms of questions. Wendy, one question we have is whether the number of genes on the custom panel would affect the statistical power for the cluster results. For example, the participant wants to know, would 100 genes be better than 400 genes to identify clusters?
Oh, no, it won't affect at all the number of genes. But of course, to identify your clusters, it depends, of course, on your what you know about the biology of your samples. For example, in this case, because I was working with mutant EZH2, I'm designing a panel based on the targets of EZH2. So I will be able to distinguish best within the B cells, the germinal center B cells, I will be able to distinguish best what are the differences between mutant and wild type EZH2. I don't know, Leandro, if you wanted to add something.
Yeah, I can add a bit on top of that. So as Wendy showed in one of her examples, the other way around probably, when we were analyzing the plasma cells and we removed the information for the Ig heavy chain, you could see that basically the different subclusters that we had identified all collapsed. So most definitely adding relevant genes can make a difference and actually can improve your clusters and the identification of subpopulations.
Okay, thank you. Can you point out what the advantage of the Rhapsody is over other single-cell RNA-seq technologies, for example, the 10-axis chromium or others?
Yeah, so the different users have also asked this kind of question. So there are advantages in using the Rhapsody, such as what Leandro just pointed out, capturing the Ig genes. That was, in our case, that was key for identification of plasma cells. And we couldn't actually detect all these subpopulations actually with the global single-cell RNA-seq. It also gives you the opportunity to look at specific transcript isoforms, as I also mentioned. Of course, you will have to do it through the custom panel design. And then based on our experience, because we did both methods, we then find new clusters using global single-cell RNA-seq. So studying less genes actually doesn't necessarily jeopardize the detection of new populations. And then I would say that the optimal single-cell RNA-seq application depends actually on the desired application. For example, for discovery of new cell type, tag counting droplet methods with high throughput are most advisable, while for analysis of allelic expression or splicing, you must choose a protocol that provides the sequencing coverage of the entire length of the RNase molecules.
Okay, thank you. Here's a question referring to one of your experiments, and that is the clustering of plasma cells. Apparently, you saw several clusters of IgG1 expressing plasma cells. And the question is, do you think those are biological differences or artifacts of the technique? And if they are biological, what's your interpretation of those different IgG1 expressing plasma cell classes?
That's a very good question. I'm also struggling with that. So for sure, what we checked is that one of the clusters, the difference of one of these clusters was like the main cluster is kappa, and there is another cluster that is lambda. And you know that in mice it's not like in human, where you have like 95% kappa and 5% lambda in mice. So there was a small cluster that actually was distinguished just for lambda expression. The other subclusters, we are not sure about that. We didn't find other obvious genes driving that subclusters, but yeah, we don't know the answer yet.
Okay, thank you. Can you say something about the bioinformatics knowledge you need for these kinds of experiments? So if somebody wants to do a single-cell RNA-seq experiment but doesn't have a lot of bioinformatics background, what would you recommend to them?
Well, this is the advantage actually of using Rhapsody. For example, I have no background, and Leandro either. We haven't. I mean, we have no knowledge about how to deal with big data actually, and this is a very accessible for users with no programming knowledge at all. Most data processing is done through an online platform, and it's a very user-friendly interface. And once you get the output files, looking at your data and analyzing using data view is very intuitive. And for users that have these big data skills, it's also very easy to import the data sets into R, and they can use their custom pipelines. I don't know, Chris, if you have anything else to add here, that you've been working a lot with the software.
I can't say anything. I mean, you've covered pretty much everything there. I would say that, yeah, data view is very easy to use and very intuitive, which is really nice. And then, yeah, the output files from data view and from the Rapid pipeline going into R or any other analysis software is very straightforward.
Okay, thank you. Wendy, are you planning to apply your observations in a clinical setting at all? And also, have you done any proteomic analysis to confirm your RNA-seq cluster results?
Yeah, so proteomics, not for now. And regarding the clinical, so we are doing now some, as I showed, some of the receptors involved in interaction with T cells were affected. And we actually have seen more consistently by protein expression the downregulation of some of the MHC class II genes. So we reasoned that we can actually combine EZH2 inhibitors with PD-L1. So we are doing, the idea is to restore the MHC class II expression in mutant EZH2 tumors, treating the mice with EZH2 inhibitors, and then start PD-L1 treatment. And we're actually doing that right now in a model where we have a tumor mouse where they express both BCL2 oncogene with the mutant EZH2. And we're on the process right now.
All right, thank you. It looks like these are all the questions we have. Yeah, I think we have no more questions. So let me thank our speaker, Wendy Béguelin, as well as Leandro Venturuti and Christopher Chin, who joined us for the Q&A, and our sponsor, BD. And as a reminder, please look out for the pop-up survey after you log out to provide us with your feedback. And finally, if you missed any part of this webinar or you would like to listen to it again, we will send you a link to an archived version by email. With that, thank you very much again for joining us for this genome webinar.